Mitosis and Meiosis by Andrew O'Brien and Tony Serrano.
Oh, jeez, man. I just woke up from this crazy dream and I was like thinking, and my cells were dividing all over the place. And I was like, wow, this is the craziest thing I've ever seen. No way this could actually happen. Don't you think that's strange?
Tony, that's not a dream. That's mitosis.
Really?
Mitosis happens all the time.
No way.
Yeah.
Are you
Your
sure?
cells are always dividing.
And why?
Mitosis is the process of dividing your cells into two identical cells with the same amount of chromosomes in them. Each of your cells has 46 chromosomes after cell division.
Maybe we should go over this.
Yeah, let's do that. Interphase is a preparatory stage for mitosis. A cell spends 90% of its time in interphase. There are three parts to interphase: G1, S, and G2. G1 and G2 are periods of cell growth. During S phase, the DNA replicates. After G2, the cell enters mitosis.
The first stage of mitosis is prophase. In prophase, the chromatin form chromosomes. The nuclear envelope breaks down and disappears. The centrioles move to opposite poles and meiotic spindles begin to form.
During metaphase, the spindle fibers are completely formed. The chromosomes line up at the center of the cell on the metaphysical plate. Each chromosome is attached to a spindle microtubule at its centromere.
During anaphase, the centromeres separate, forming sister chromatids, which are pulled to opposite poles of the cell. The cell now has double the chromosomes than it originally had. Any chromosomes without a centromere will be left behind.
Telophase and cytokinesis are the final stages. Two new nuclei form around the chromosomes at opposite ends of the cell. The chromosomes begin to unwind, resembling interphase. Accompanying telophase is cytokinesis, in which a cleavage furrow forms, splitting the cell into two identical diploid daughter cells. The importance of mitotic cell division is to renew and repair cells and for growth and development.
Oh man, I didn't even know that was what was going on in my body. Is there anything else I should know?
Well, there's another process called meiosis.
What exactly is meiosis?
Meiosis is another type of cell division. There are some key differences between meiosis and mitosis. Mitosis only has one cell division, while meiosis has two cell divisions. Synapsis of homologous chromosomes occurs in meiosis. This creates genetic variation, while mitosis creates cells that are identical. Mitosis creates two diploid cells, while meiosis creates four haploid cells. Just keep in mind that meiosis occurs in two parts: meiosis 1 and meiosis 2. In meiosis 1, DNA replication occurs, creating homologous pairs which are separated into two cells. In meiosis 2, homologous pairs are separated into sister chromatids, creating four haploid cells. I'll show you exactly how this is done.
The first stage of meiosis is prophase 1. During prophase 1, the chromosomes begin to condense. Synapsis, the pairing of homologous chromosomes, occurs. During synapsis, crossing over, the exchange of corresponding segments of DNA molecules by non-sister chromatids is completed, resulting in each homologous pair having at least one chiasmata. The chiasmata is a point where crossing over has occurred. Centrosomes move to opposite poles, spindle fibers form, and the nuclear envelope breaks down.
In metaphase 1, the pairs of homologous chromosomes are arranged along the metaphase plate. Both chromatids of one homologous pair are connected to one microtubule from one pole. The other homologous chromosomes are connected to a microtubule from the opposite pole.
In anaphase 1, the homologous chromosomes are pulled by the spindle fibers to opposite sides of the cell. The sister chromatids move as one unit towards the pole.
In telophase 1, each side of the cell has a complete haploid set of replicated chromosomes. These chromosomes are genetically diverse due to crossing over. Cytokinesis occurs, creating two separate haploid cells with replicated chromosomes.
In the middle of the night, I go walking in my sleep.
During prophase 2, the spindle fibers reform and move to opposite poles. The chromosomes begin to move towards the metaphase plate.
In metaphase 2, the chromosomes line up along the metaphase plate. The two sister chromosomes are not genetically identical due to crossing over. Each chromosome is attached to a spindle microtubule at its centromere.
During anaphase 2, the breaking down of proteins at the centromere allows chromatids to separate. The chromatids move to opposite poles of the cell. Each set of chromosomes is now considered a haploid, without a replicated chromosome pair as they move to opposite poles.
Finally, during telophase 2, the nuclei begin to reform, and the chromosomes decondense. Cytokinesis occurs, producing four not identical haploid cells. The importance of meiosis is reproduction with genetic diversity.